Thank you for the introduction. Hello, my name is In Lee, and I am a researcher and business development senior manager at SHIFTBIO. SHIFTBIO aims to lead a new paradigm shift in drug development using our natural drug delivery platform. To start off, I want to explain why we are interested in using the natural drug delivery system. For decades, scientists have discovered many targets to fight against diseases. Unfortunately, the current ways we make drugs, like using small molecules or antibodies, only works for less than 5% of these targets. This means that there is a huge number of undruggable targets. What this fact tells us is clear. We need a promising drug delivery system to make undruggable targets into druggable. To address these challenges, we're using natural nanoparticles. These NMPs are produced from all cells that evolved over millions of years to shuttle biomolecules into human cells. And of course, we believe that NMPs have great advantages over current drug delivery system. For example, NMPs have unique abilities for extrahepatic delivery, crossing the biological barriers including the BBB, and the direct delivery of cargoes to target cells by avoiding endolysosomal pathway. There are over 140 global clinical trials of NMP therapeutics, with some advancing to phase 3 in US FDA. This evidence shows the feasibility of NMP therapeutics in the near future. So what makes us different? We are different because we have developed drug engineering scaffold, the NMP sorting motif, or the NSM. The NSM refers to a specific amino acid sequence that can be selectively transported to NMPs during the biogenesis and loads cargoes linked to NSM into NMPs. And this is a platform technology to engineer NMPs to deliver undruggable cargoes. Our NSM platform technology has three distinctive advantages. First is high cargo loading efficiency. Second is multiplex cargo loading, meaning that we can load therapeutic cargoes on the outer side of NMPs or the inside or at the same time. The last one is universal cell applications. Our NSM platform technology has a broad applications to not only HEK hex cell lines and also stem cells, including mesenchymal stem cells. From now on, I want to explain why we must use our platform technology to create the next generation medicines using three examples. The first example is a membrane protein. The activity of membrane proteins depends highly on the transmembrane domain, or TMD. Our NSM platform enables expressing membrane protein on the NMP surface by maintaining the TMD. For example, we developed SIRPA NMP, which highly expresses SIRPα alpha membrane protein on the surface, and have found that NMP provide the optimal microenvironment to the function of SIRPα alpha protein compared to other drug delivery systems. Furthermore, we have confirmed the higher efficacy and safety of SIRPA NMP over CD47 antibodies. The second example is transcription factor. Transcription factors are master regulators of multiple genes. Even though transcription factor has great therapeutic potential and application in diverse diseases, it is nearly impossible to directly deliver transcription factors to target cell's nucleus. However, our NSM platform technology enables in vivo delivery of transcription factors. We first found that naive NMPs can cross the BBB and deliver Cre protein into the targeted cell's nucleus very efficiently. Next, we successfully loaded transcription factor, or the HIF-1 alpha, into NMP using our NSM platform and have confirmed higher loading efficiency compared to current natural scaffolds such as CD8 and CD9. To leverage this finding, we have developed a strategy that allows in vivo cell reprogramming. We have confirmed that HIF-1 alpha NMP can specifically target immune cells and promote reprogramming through the transcription factors. The third examples are RNA. As you know, RNA is composed of either small RNA such as ASO and siRNA and large RNA such as mRNAs. we have different loading strategies to each RNA cargos. I want to first start with that many scientists in the past thought that small RNAs are located in the inside of NMPs. However, recent research and our findings suggest that most small RNAs are associated with the NMP surface. Naturally, NMPs can deliver small RNAs by a distinct cellular uptake mechanism, even when small RNAs are associated on the outer side of NMPs. We simply leverage this natural phenomena and anchor small RNAs on the surface of NMPs. And by doing so, we have confirmed a sufficient therapeutic efficacy. However, in the case of an mRNA, we need a different story. We need a different approach. If we were to anchor large RNAs to the outer surface of NMPs, this may cause unwanted immunogenicity and adverse side effects. Therefore, we have to find a way to load this mRNA into the NMPs. And our SHIFTBIO team was first to identify the specific NMP fraction that can load mRNA efficiently when targeted mRNA is overexpressed. We have secured our IP right regarding this finding. And if you look in the bottom two figure, we have loaded different types of mRNA into specific fractions of NMPs. On this slide, I want to explain how we are using our NSM platform to enhance the intracellular delivery of the RNA cargos to target cells. Most of the audience here will appreciate that the important fact about RNA delivery is not the loading efficiency itself, but how well we can avoid the endolysosomal pathway to deliver our RNA cargos. Regarding this point, we have two different strategies. The first strategy is using fusogen. If we can express the fusogen on the NMP surface using our NSM platform technology, we can induce a fusion mechanism and thereby avoiding the endolysosomal pathway. We have comprehensive IP regarding diverse fusogens such as GSVG to RSVF and GALA fusogens. The second strategy, and this is a very recent finding, very interesting finding from our group, is that we can use a different, if we are to express peptides on the NMP surface, that we can induce clathrin-independent endocytosis. As you know, most NMPs enter the recipient cells through clathrin-independent endocytosis, which almost degrade cargos by endolysosomal pathway. However, if we express a certain peptide that can induce clathrin-independent endocytosis, we can avoid the endolysosomal pathway and deliver our RNA cargos very effectively. We have a POC data using KRAS siRNA showing that clathrin-independent endocytosis NMPs can effectively deliver the small RNA cargos compared to the commercial lipid nanoparticles and naive NMPs. So thanks to our platform technology, we have developed various programs. However, today I want to focus and introduce our first program, SIRPA NMP, which has been designated as the orphan drug designation by the US FDA. The SIRPA NMP is isolated and purified from engineered stem cells, MSCs, and have dual mode of actions. First mode of action is using the regenerative factors of stem cells. The second one is blocking the CD47 that is highly expressed on pathological fibroblast or necroptotic cells by using SIRPα alpha protein. We have seen in vitro that SIRPα NMP selectively targets CD47 ligands, and in the in vivo data using biodistribution, we have seen the SIRPα NMP preferentially and effectively accumulates to CD47 overexpressed tissues. Furthermore, we have corroborated this data by pre-treating CD47 antibodies prior to our NMP. As you can see, the SIRPA NMP has decreased when the CD47 overexpressed tissue was pre-treated with anti-CD47. Furthermore, compare in case of naive NMPs and SIRPA NMPs, they both effectively target immune cells. However, for the SIRPA NMPs that went beyond immune cells and specifically target hepatocytes, which in this case overexpress CD47. Next, we have confirmed the toxicology and toxicity of the SIRPA NMP. In this study, we used a dosage that is 100 times higher than our therapeutic dosage and does not confirm any adverse side effects. And furthermore, we have seen the great therapeutic efficacy of SIRPA NMPs in APAP-induced models, APAP-induced acute liver models, which is the leading cause of acute liver failure in the United States. The data not shown here, but we have confirmed the therapeutic efficacy in diverse models that recapitulate different etiologies, including LPS models, TAA, and CCL4. And furthermore, I want to emphasize the only current proven drug, FDA-approved drug is NAC. However, SIRPA NMP showed a greater therapeutic efficacy compared to NAC. Next, we found that CD47 is also overexpressed in fibrotic diseases such as IPF and NASH and liver fibrosis, and confirmed the anti-fibrotic effects of SIRPA NMPs in diverse fibrotic models. From now on, I want to switch my gear, and talk about our CMC strategy. So we're very confident in our manufacturing process. We have developed our manufacturing process with a US-based company named SHIFTBIO. With collaborating with SHIFTBIO, we have developed very rigorous upstream process development and downstream process development that ensures high purity and high yield. To start with, we first engineered and developed our stably transduced master cell banks using human bone marrow-derived mesenchymal stem cells. The first fact we checked was whether these STMCBs maintain the characteristics of MACs. And furthermore, we have seen the intra-donor variability and inter-donor variability. Next is our upstream process development. In this section, I want to emphasize that we have seen a scalability between 250 milliliter, 15 liter, and 50 liter between 3D bioreactors. And compared to 2D cell culture, 3D bioreactors generated higher yield. We have seen batch-to-batch consistency in diverse batches. For the downstream process development, we have 6 different unit operations where we have optimized each unit operation to ensure the highest recovery rate while removing as much as impurity as possible. So as you can see, we have achieved a recovery rate up to 50%, while the protein contaminant clearance was 96%, host residual DNA, such as dsDNA, was removed with 93%, and human protein surrogate, we measured albumin as to see, and the 99% was cleared. Until last September, we are in a stealth mode. But this year, we're actively promoting our technology, our company. So until now, we have various meaningful milestones. One of them would be we have secured $10 million USD of non-dilutive fundings. Our company was founded by 3 MD PhDs, meaning that we have a high and great understanding, in-depth understanding of the high unmet medical needs. Our team, our SHIFTBIO team, we have 23 members who are willing to dedicate their lives to make better medications, better therapeutics. And our scientific advisory board, we have great scientific advisory board from experts of their respective fields. So long story short, we want to bring the next paradigm shift using our natural drug delivery system to make the impossible possible and the undruggable druggable. Thank you for your attention. Thank you for listening.